Hi everyone. In today's video, we're going to talk about the connection between meiosis, protein synthesis, and genetics. And we're going to look at it through the idea of ladybug love. So this is the idea of what we're going to be covering today. So we're going to do a short, really simplified version of meiosis. We're not going to go through all the steps. I'm going to model the fertilization process. We're going to look at the alleles that are passed on to the offspring and interpret what proteins are produced and how it affects the appearance of the ladybugs. Just a note, this is not meant to teach you about ladybugs. This is all made up stuff. It's not like true to how many chromosomes the ladybug has, okay? It is just meant to understand this process in biology. So we're going to pretend that our ladybug just has 5 chromosomes. Okay, so I colored these pink because these are the chromosomes that the mother ladybug has. Again, they're homologous chromosomes, which means that they're the same genes. Homo means same, so they're the same genes on both sets of chromosome number 1, but one was from this ladybug's mom and one was from her dad. Now, what's going to happen is that when she makes her eggs, it's going to be random which of these chromosomes goes into each egg. So I'm going to cut this apart to model the next process for you. So here we've got mother ladybug's chromosomes, and you can see that she's got normally 5 sets of chromosomes for a total of 10 chromosomes. Now, during the process of meiosis, or egg formation, we're going to have to reduce this in half so that the egg has half of the DNA and the sperm has the other half. So when the baby's formed, the baby gets a full set of 10 chromosomes, like is shown here. So we're going to represent this creation of the eggs with, I'm going to use the help of a coin. I'm going to flip this coin. See, it landed on heads. So that means that for chromosome number 1, I'm going to pretend that when the egg formed, this is the chromosome that was put into the egg. Okay. Chromosome number 2 also landed on heads. You will notice it was random which of the number 1 chromosomes ended up inside the egg. Okay, and because I flipped heads this time, did not impact whether I was going to flip heads or tails the second time. So basically, they sorted independently of each other. It's called independent assortment. So here is one egg created by the mom. We're going to now do the same process with dad's chromosomes. Here is the father ladybug's original DNA. He's got 10 chromosomes, 1 copy from mom and 1 copy from dad for each of the 5 pairs of chromosomes. I'm going to cut them apart so we can simulate sperm production. Here are dad's chromosomes. We're going to now flip to see which chromosome number 1 is passed on to his sperm. These are what is going to be in the sperm, and here are the chromosomes that are in the egg. So when the sperm fertilizes the egg, we're going to end up with a genetic combination like this. Just like before, we'll see baby has 1 copy of chromosome number 1 that they inherited from their mom, and 1 copy of chromosome number 1 they inherited from their dad. Here we've got a good example of homologous chromosomes. You'll see that they have the same gene, so that's where the homo part of the name comes from, homologous, same gene, but they're going to have a slightly different DNA sequence. have labeled those different DNA sequences with a capital D or a lowercase d. And this is really to show, we call it an allele, which is a particular version of a DNA sequence. And that's going to impact what protein. This is going to make a different protein than that. Okay, so I just took the information that we just saw with our baby's chromosomes and I put it into a chart. So for each chromosome number, I recorded the DNA sequence, again, that we had been calling an allele, because it's a version of that DNA sequence that came from the egg, the one that came from the sperm, and then what would the combination be for baby. What I have done is that for each time there's a capital letter, what we are indicating is that there is a functional protein produced. So, for example, since the baby had a capital A for color of wings, that means that that baby was able to produce a red protein. And so the baby will be red, okay? If the baby had only inherited little case a's, that means that baby would be white in color because the protein was broken. So there would be no red coloring produced, okay? So far we're gonna have a red wings and oh, look at this. We've got a protein induces the growth of one extra set of legs. So we're going to, in this case, say that the DNA sequence caused the baby to now have 8 legs instead of 6, which is standard for ladybugs. This is interesting. Here we have one functional protein and one broken protein. And as long as you have one functional protein, that means that protein will still be produced by the baby. So that means, cool, we're going to have some blue spots on our ladybug. Both DNA sequences coded for extra antennae. Okay. Oh no, look, now we've started to have a lot of DNA sequences that coded for broken proteins. The eye spot on the head is not going to be produced, and the color of the head is going to be white, and the color of the antenna are also going to be white. So what do you think would happen if we were to do it again, and again flip coins to see which chromosomes ended up in the egg and which chromosomes ended up in the sperm? Let's find out. So here is our DNA combination for the new offspring, the new fertilized egg. Okay, let's see how this baby maybe is going to look different than the last one. All right, so it looks like, again, baby's going to be red in wings and have an extra set of legs. There's still going to be blue spots. We're going to have an extra set of antennae. F, we have a one capital F, so that means we have one allele that produces a protein. So that means that we would have green eyes. That's different. And in G, we got 2 lowercase g's, which means we have 2 broken copies of that DNA sequence. It does not code for a functional protein, so there's no extra eye spots. And likewise, we only have non-functional proteins at these locations on the chromosome. So you could see that each time we would go through this process, we're going to end up with a baby that looks different. So if we were to draw out these baby bugs, each one would be a little bit unique. And this activity that I just showed you does not even take into consideration crossing over, which is another way that additional genetic variation becomes possible in the production of offspring. And so why there's such a rare chance that any 2 siblings would be identical to each other. So I hope this video helped. You now understand a little bit about meiosis and the importance of independent assortment in the process of fertilization, and that those alleles mean that really it's a DNA sequence that codes for a protein, and whether or not it's a functional protein, we're going to see a difference in the bug's appearance. Please give a thumbs up if this helped you, and subscribe to find more videos like this. Have a great rest of your day. Bye-bye.